very much for that very nice introduction. And Hampus, I give the word to you.
Hi everyone, and most welcome to my PhD dissertation. So I've spent the last 5 years to develop instrumentation for measuring vegetation and insects. Because the focus of these instruments, as Oda said, is to make them as accessible and as cheap as possible. And to do this, I've built them using 3D printers and Lego kits. So throughout this presentation, I will show you how I made this 6-dimensional camera that we talked about. I will tell you how I made a device that can measure fluorescence signals from 100 meters away. You will also learn how to tell a mosquito, a male and a female mosquito apart, and how to measure the insect's wing thicknesses while they fly. And before I get started, I want to thank all my collaborators, all my co-authors, my funders, and especially Professor Bodovic for this great introduction. And, I'm really impressed you managed to cover it so well in such a short amount of time. So I'm very grateful for that. So, why should we care about insects and vegetation? Well, they're all around us and they impact our lives in countless different ways. Vegetation is essential for life on Earth and it's more fragile than ever due to increasing climate changes. Insects, they are a huge part of our biodiversity. They constitute more than half of all known species, and this includes all types of animals, plants, fungi, bacteria, you name it. Still, insects are is 52% of all species that we know. It's quite incredible. And from a human perspective, we can divide insects to different groups. Insects can serve as pollinators, and in fact, insects pollinate around 90% of all flowering plants. Again, quite incredible. On the other hand, there are other insects that are not as helpful to us. Pests can cause large crop failures, which can lead to starvation. And other insects spread diseases. The mosquito kills more humans than any other animal by spreading diseases such as malaria, yellow fever. And they kill more of us than we manage to do ourselves, including all war and violence. We still only come on the second place after the mosquito. So to monitor insects and vegetation, it helps us to understand processes, which in turn can help us combat diseases, hunger, and climate change. So how can we monitor the biosphere? Conventional methods, they typically require a lot of trained manpower. So you have sweep nets, or you place out traps, or you have quadrat surveys like this, and you collect your samples. These samples can later be studied, either with visually, with microscopes, or with DNA sequencing. So with these kinds of methods, they are useful, but it's hard to get a high resolution in time. You can't empty your trap every minute of the day. And it's also hard to get a good resolution in space. You can't place these traps in every square meter of forest. But there are other more advanced alternatives, such as gluing antennas onto bees and then tracking them with lidar. Looks a bit strange, I agree, but this is the thing, I promise. Or you can do hyperspectral imaging, as we saw saw, from satellites or from airplanes to monitor vegetation. So as you understand, these types of methods are usually both complicated and expensive. So is it a problem that it's expensive? Let me highlight one example. So here is a map which quantifies how much pollinating insects contribute to the economy. So here green means that we have little contribution by pollinators to agriculture, and orange and red means that they contribute a lot. So if we just look at some examples, cases here. So in Ghana, in Italy, and in South Korea, we all have these orange and red zones. So here insects are apparently important. But does this mean that in the green areas that insects are not important there? Before we draw any conclusions, we can have a look on another map. And here, this map instead shows how many insect studies are done in different regions. And it just so happens that in these exact places where we saw a lot of that insects contribute a lot, that this is where we monitor insects a lot. So it turns out the more you investigate insects, the more they seem to contribute. And this shows that we don't have a full understanding of how insects contribute to a large part of the world, and especially in tropical regions like sub-Saharan Africa, which is also very important. These are regions with huge biodiversity that we don't know much about, and also regions that rely heavily on agriculture. So it is of great importance to monitor our biosphere all over the world. And to do this, we need accessible instrumentation. And this brings us back to the title of the thesis, and this table that you've already seen with different papers. So toward accessible biophotonics. During my thesis project, I've produced 11 different papers that cover low-cost instrumentation, described in papers 1 to 4. And I've described their application within insects and within vegetation in papers 5 to 9, and some perspectives on the instruments used in education and in accessibility in papers 10 and 11. So if we switch out the titles of these papers to some buzzwords, this is how it could look. So throughout this presentation, I'm going to go through a few of these papers more thoroughly. So I will describe the instruments in papers 1 and 2 called Biospace and Fluorescence lidar in quite some details, because this is where I think my main contributions lie. And after that, I will go a bit briefer through some results. I will show you remote wing measurements and how to tell mosquitoes apart in paper 3 and 4. After that, we will take the instruments to challenging conditions in Africa and look at dusting bees with fluorescent powder in paper 5. We will look at daytime fluorescence of leaves in paper 8. And finally, we conclude with the lasers in the jungle in paper 6. So, in the first paper, we made this instrument called Biospace, which we say is a 6-dimensional camera. So what can I mean by that? First, we have to discuss what is a light measurement. And when you do a measurement with light, you typically have a light source, and you have a detector, and you have a target. So you illuminate your target, and you record the light that comes on the other side. And this light, it has different properties. Light has an intensity, a position, you have a propagation direction, some wavelength, light has a polarization state, phase, and some temporal modulation. And by comparing these properties before and after they interact with the target, you can learn something about the target. And there is a lot of interaction processes when light interacts with a tissue, with a target. And by knowing the theory behind these interaction processes, we can explain what makes up this target. So let's do a short crash course in how light can interact with a tissue. So let's say an insect body in this case. So if light comes in to the insect, it the insect has a different refractive index, so you will have some reflection and you will have some refraction of the light. And then inside this insect you will have different particles that can absorb the light and so on. So the light can be scattered, it can be absorbed and re-emitted as fluorescence. And here you can note that the line here changed color, and that's because fluorescence changes the wavelength of the light. Now it hits the boundary again, so you will have more refraction and more reflection until the light is finally absorbed in the tissue. So, by knowing the theory behind these interaction processes, we can investigate targets. So now we need an instrument that we can compare these light properties before and after these types of interactions. And here comes the instrument described in Figure 1, called Biospace, which stands for Biophotonics, Imaging, Optical, Spectral, Polarimetric, Angular, Compact Equipment. So we've said that it can measure in 6 dimensions, and we're talking about these light properties. So it can measure 3 spatial dimensions. It can measure the polarization of light. It can measure scatter angle, and finally the wavelength, so the color of light. And to do this, we mount the target on a rotation stage. And we have here a camera in the bottom of the image, and we have a light source on the right side of the image. And the target is here spinning around. So by spinning it, we can get information from different angles. And then, to get the different scatter angles of the light, we rotate the light source around. And to get different polarization states, we have a polarization filter in front of the camera. So this covers the first 5 dimensions. And to get the wavelength, we've designed a specialized light source. So this works on a principle called LED multiplexing. So this light source, it has 8 LEDs with different colors, different wavelength bands, and it's synchronized with the camera. So with each image that the camera takes, the sample that we have is illuminated by one of these LEDs. And then the camera takes another image, and we illuminate the sample with the next LED. And it goes on like this, so that we take these 8 different images and one dark one, and then we have built up a multispectral image of our sample. And the result, it can look something like this. So here you have a bark beetle shown for 3 specific colors as red, green, and blue. And to your left side, you have it in copolarized light, and on the right side, you have it in depolarized light. So copolarized basically means that we're only looking at the same polarization that we illuminated with. And depolarized light means that we're looking at only the light that has flipped polarization state. And here we can learn specific things about the targets that we later can use when we design our lidars and so on. So, for example, here we could learn that when we look in the copolarized light, we have these bright flashes from the wings, and you can see these soap bubbly looking colors that we talked about before on this left-hand side. While on the right-hand side, when we're looking at the depolarized light, you don't have any flashes. But instead, you have this glowing look of the body. And this is because now we're looking at the depolarized light, so the light that has flipped polarization state. That is the light that has scattered multiple times inside this insect's body. And we can use this later. So in paper 4, we use these soap bubbly flashes to determine the wing thickness of some flying insects. And we use these copal and dipole light in the lidar in paper 6, where we use this polarization information to classify insects in the jungle. So that was Biospace for the first paper, and now I'm going to explain the fluorescence hyperspectral lidar, paper 2. So I say say lidar, and lidar stands for light detection and ranging. So it's something that can measure distances reached with light. Typically this is done by sending out a pulse of light, then some light comes back and you count the time it takes. By knowing the time, you know the range. Our lidar works in a completely different way. We're using something called triangulation. So we have a laser that comes out, we have a lens and a detector, and we tilt them to different specific angles so that one part of this laser beam is imaged to a specific part of the detector. So if you read out 1 pixel of this detector, you get the signal from this part of the laser. So this is called a Scheimpflug lidar, and it's we use. But I said that this is a hyperspectral Scheimpflug lidar, or hyperspectral lidar. So this becomes a bit more complicated. So now, instead of this detector, we have a slit. So I'm going to clean up all of these lines. So we replace the detector with a slit. So now the light will travel through that slit. And we can image it again onto an image sensor over here, using the same criteria that we used before. And again, we will have distance on this image sensor. So near and far will represent different parts of this beam. But in between, we have the disperser. So the other axis of this image sensor is used to register different colors of the light. And that's the principle. And to build it, we designed it with ray tracing software. And that can be used to find the perfect optimized distances between the components. And when we have that, we can design a 3D structure around it with CAD software and 3D print it. So this is a way to cut down on costs. Then we're not only using 3D printed plastic, we also add some other components for alignment purposes and so on. So here's the full view of the lidar. And the laser comes out, it's collimated and with polymer, comes out this way. And then the light that comes back goes through this receiving lens and hits the camera again. So the data that we get from this lidar is in the form of a data cube. So one axis of the sensor has the range information, the other axis has the wavelength information, and then we take multiple images to get signals. So in this data cube, we can measure distances from 5 meters to 100 meters. We can measure wavelength from 400 nanometers to 810 nanometers with resolutions of 6 nanometer, and we have range resolutions that varies on the distance. So it goes from a few centimeters when you're close to almost a meter when you're far away. So we wanted to try and see that this lidar works. So to do that, we did one experiment where we had a couple of insects and we fed them with fluorescent sugar water. And this mosquito right here drank a lot of this sugar water and probably obtained some sort of superpower from it. Yes. So we had it into the lidar, and sure enough, we got this green blob you see here is the fluorescence from this mosquito, the green Hulk mosquito. So it seems to work. So that is the two main instruments that I'm going to talk about. Now I'm going to show you briefly the instruments in paper 3 and 4, but they are based on the same principles. So the lidar in paper 3 is very similar to the lidar you saw before, but here instead we're using longer wavelengths. And this can be used to look at wing thicknesses of insects. So in this case, when you have a wing and you get these, the signals back, you get a spectrum. So for different colors, you have high intensity. For other wavelengths, you have lower intensity. And if you know the theory behind it, you can fit a model to it, and you can estimate the wing thickness. So for your reference, a damselfly wing is around 1.4 micrometers. Now you know. We tested this outdoors near Lund, and it gave signals from 90 meters away. So this is a photograph of the lidar is in this tent to the left side, and the beam is coming out. And you can see that there's a flash here, and that is because there was an insect that flew into the beam as this photo was taken. So for paper 4, we used multiple instruments, and one of them was a modification of Biospace, where we basically turned it upside down because now we're having hanging targets. So in this paper we're looking at mosquitoes, and we also switched out the wavelength range. So we're looking at longer wavelengths. We have a different light source and a different camera. And we did different measurements, but with the hyperspectral images of mosquitoes that we found, it's possible to estimate the optical path length of the different absorbing compounds in the mosquitoes. So for mosquitoes in this wavelength range, it's mainly melanin that we have in our skin and water that absorbs most of the light. And we can find out by illuminating these mosquitoes how much melanin and how much water does the light travel through. And by knowing this, we can differentiate between, here we have 3 cases of female mosquitoes. These are Aedes aegypti. They're really nasty ones that transmit Zika and dengue fever. So you can see that there is difference between the spectra from these groups, and that is coming from that they have different water pathways in this group. So that's how you can tell mosquitoes, male and female mosquitoes apart. If you don't have access to hyperspectral instrumentation, you can actually look at their antenna. So if they have a fluffy antenna, it's a male mosquito. So now I talked about the instruments. I talked about some measurements in Sweden. And now comes the true test of accessible instrumentation. Let's see if we can deploy it in challenging conditions. So for paper 5, we bought a lot of components needed to replicate Biospace and this fluorescence lidar, and we went to Ghana. So I brought a lot of Lego, and I brought a 3D printer on the plane and went there. So this is the 3D printing of half of the lidar, and this might look fast and easy, but in reality that printed 10 hours. And this was the 3rd try. Because the first 2 tries we had a power blackout, so we had to throw away the pieces. So there are some challenges. But we managed to recreate both Biospace and the fluorescence lidar in their lab in Ghana. And then for this study, we packed up the lidar. We drove out to the countryside to a place where they do research on stingless bees. These are bees that produce honey, but they don't have a stinger, so very nice. So as we put up the lidar, we noticed that there was another big blackout, and but we managed to find a generator, so we could start with our measurements. So the idea of this study is that we have this lidar set up next to these stingless bees, and we put fluorescent powder on the entrances of these bees. So the ones here are slightly bigger bees, which we had some green powder on their entrance, and then we had red powder on the entrance of these smaller bees. The idea is that when they fall out of their hive, they get some powder on their body, and then when we detect them with the lidar, we can see that they have this fluorescent signal. So as you remember, we have this data cube that comes from the lidar. So in this case, it's a 2-second measurement, and you can see this purple cylinder here. It's at 22 meters away, and it's there for the whole time. So that is basically where we're aiming the lidar at. It's called its determination. But then there's a bunch of different spots. These are insects that's flying into the lidar. And with some of these insects, we also have a fluorescence signal. So this one is a red tag bee, and that one is a green tag bee. So by taking 26,000 of those data files and analyzing them, we found 18,000 insects over those 24 hours that we measured. And with that, we could make some statistics. So this is how it looks, where you have range on the y-axis, and you have time on the x-axis. So you can see on the graph in the bottom there, that, for example, we can see that these red tagged bees were very active in the morning and the evening, while they were not flying so much in the warmest hours of the day. While the green tagged ones, they were active throughout the day. So these are things that we can, the types of things that we can learn by this type of measurement. And let me note something which makes these studies stand out. So here we monitor fluorescence signals during 24 hours, which means during full sunlight. So fluorescence is well studied and it's used a lot, but mostly it's done in dark laboratories. Or when you do it outside, you do it during nighttime when it's dark. This is because fluorescence is typically very weak compared to the sunlight. But if you want to measure bees like this with fluorescence, you really have no choice. You have to measure them during the day because they are day active. So, so you have to have an instrument that can measure daytime fluorescence. And this is the demonstration of the first demonstration of this type of measurements that we have seen. So for the next paper, it's during the same trip to Ghana and the same workshop. We wanted to see if we could do the same with leaves and canopy. So can we do daytime fluorescence there as well? So we placed the lidar on top of a large building, looking down at this forest canopy where we could scan. And sure enough, we managed to get fluorescence signals both during nighttime and during daytime. So these fluorescence signatures that you get can be used for finding out which species it is or the health status, while you can get this canopy structure from the range information. So in the same study, we did remote measurements of a bunch of different leaves for looking at their specific fluorescence signatures. And you can find that they are slightly different for different species, and especially for different health status. So this is the final paper that I'm going to dive into, and in this case we wanted to push up the accessibility up even more. We wanted to crank it up to 11. And to do this, we decided to go to a virgin rainforest in Ivory Coast. And it took us 3 days to get from the airport all the way into the jungle with all the equipment. But we got there, and we packed up the lidar into a tent. We had it all powered by a generator, which only broke down a few times. And we collected a bunch of insects that we measured inside that cardboard box over there, which of course contains Biospace that we brought to the jungle. So to explain our experiment, I took a bunch of photographs with a drone, and I made a 3D reconstruction of this experimental site, which you might recognize from the front cover of the thesis. So in order to explain the experiment, I made an animation as well. So we had the lidar here by the forest edge, and then we moved it to different angles so we can go different heights. And then inside the forest, we also had different traps. So up there in the height of the canopy, we have some traps, and down at the bottom as well, and a zip line trap which covers different heights, all different heights of the canopy. So the idea here is that we want to probe these different heights of the rainforest, because a rainforest has these different structures, it has these different layers of vegetation. We want to see how that can impact the insect activity at the different heights. So so it looks something like this. We have these different laser angles, and here is some of the results. So here each dot is one insect that we measured, and the color of that dot is the time of day when it was measured. So here you can just see that there's a bunch of insects near the ground. But if you look at the color, it's a lot of blue and green. So apparently morning insects and nighttime insects, they really like hanging by the ground. Well, during the warmest hours of the day, like orange and red, they're avoiding this area and instead hiding a bit more in the shady parts of the canopy, which is 25 meters tall. So this concludes my talk. I've briefly gone through much of my activities the last 5 years. I have learned a lot, and I've had a lot of fun as well. So I hope this presentation has been both entertaining and enlightening for you.